Good afternoon, everyone. Thank you for attending my talk today. My name is Tisha Bhatnagar, and today I will be talking to you about my exciting summer research project in Dr. Seth Blackshaw's lab. The title of my talk is Investigating the Effects of Growth Factor Pathways on Tanycyte Proliferation and Transdifferentiation in the Postnatal Hypothalamus. The hypothalamus is the master regulator of major physiological functions, such as metabolism, feeding behaviors, sleep, and circadian rhythm. In the hypothalamus, radial glial cells called tanycytes line the wall of the third ventricle. Tanycytes detect hormonal cues in the cerebrospinal fluid and can communicate these chemical signals to nearby neurons using a single basal process. Tanycytes also express genes similar to neural stem cells. In recent years, evidence now supports that tanycytes have the potential to generate neurons during postnatal development. Recently, the Blackshaw lab has demonstrated that tanycyte-specific loss of the nuclear factor 1 family of transcription factors, NFI-A-B-X, leads to an increase in tanycyte proliferation and transdifferentiation into neurons. Tanycyte-derived neurons fire action potentials and can respond to hormonal cues such as leptin. Single RNA sequencing analysis shows an upregulation and downregulation of various growth factor pathways following the loss of the NFIs. The Blackshaw lab has also shown that pharmacologic activation of the Wnt pathway in NFI-deficient mice enhances tanycyte proliferation and transdifferentiation in the parenchymal cells. During development, growth factor pathways play a critical role in neural stem cell proliferation and transdifferentiation into neurons. For example, the Wnt pathway is known to promote proliferation and neurogenesis, while the Notch pathway is known to inhibit proliferation and neurogenesis. Based on these studies, my research question is, does the targeted modulation of the Wnt and Notch pathways stimulate wild-type tanycyte proliferation and transdifferentiation into hypothalamic parenchymal cells? My experimental outline is as follows. I first injected wild-type mice with tamoxifen at postnatal days 3 through 5 to activate Cre in tanycytes, leading to nuclear GFP labeling of tanycytes. This also allows us to label and track parenchymal cells derived from the tanycytes. Next, on postnatal's day 10, I injected mice with either vehicle or ABC99 for 3 days. In addition, I also injected EDU for 3 days to determine if ABC99 stimulates tanycyte proliferation. 4 days after the last injection, I performed immunohistochemistry and cell counting. In the vehicle-treated mice, I observed baseline levels of tanycyte proliferation, shown with the blue arrows, and transdifferentiation, shown with the orange arrows. However, in the ABC99 treated mice, I observed an enhancement of tanycyte proliferation and transdifferentiation into parenchymal cells. Here, I am showing GFP-positive parenchymal cells derived from tanycytes with orange arrows, GFP-positive EDU-positive parenchymal cells with red arrows, and GFP-positive EDU-positive tanycytes with blue arrows. When I quantified this phenotype, I observed an increase in the number of GFP-positive cells in the hypothalamic parenchyma in the ABC99 treated animals. I also observed an increase in the number of double-labeled GFP-EDU-positive cells located in the parenchyma in the ABC99 compared to the vehicle-treated animals. I didn't observe a significant difference in the number of EDU-positive tanycytes between the vehicle and ABC99 treated mice. However, I will need to follow up with more biological replicates. Based on these preliminary results, my data suggests that acute Wnt activation in wild-type tanycytes stimulates tanycyte proliferation and transdifferentiation into parenchymal cells in the postnatal hypothalamus. Next, I examined whether Notch inhibition in wild-type tanycytes also stimulates tanycyte proliferation and transdifferentiation. As stated previously, Notch signaling is known to inhibit proliferation and neurogenesis. Similarly, I injected tamoxifen at postnatal day 3 through 5. To inhibit the Notch pathway, mice were injected with a gamma secretase inhibitor LY 411575 at postnatal days 10 through 12. EDU was also co-injected and immunohistochemistry was performed 4 days after. Again, in the vehicle-treated mice, I observed baseline levels of tanycyte proliferation, shown in blue, and transdifferentiation, shown in orange. Interestingly, when I inhibited the Notch pathway in wild-type tanycytes, I observed a decrease in the number of GFP-positive cells in the parenchyma compared to the vehicle-treated animals. I also observed a substantial decrease in the number of double GFP EDU-positive cells in the parenchyma compared to the vehicle-treated mice. When I quantified this phenotype, I observed a reduction in the number of GFP-positive parenchymal cells compared to the vehicle controls. There's also a decrease of transdifferentiated parenchymal cells labeled with EDU. Overall, this data suggests that Notch inhibition potentially suppresses tanycyte proliferation and transdifferentiation during postnatal development. This is different from what is known about the Notch pathway during embryonic development. In summary, I've demonstrated that acute activation of the Wnt pathway in wild-type tanycytes potentially stimulates tanycyte proliferation and transdifferentiation into parenchymal cells, while acute inhibition of the Notch pathway suppresses wild-type tanycyte proliferation and transdifferentiation. Since our goal is to stimulate tanycyte-derived neurogenesis, my future experiments will include performing immunohistochemistry to see if acute Wnt activation stimulates the generation of tanycyte-derived neurons. I also plan to perform chronic Wnt activation and Notch inhibition in order to enhance the subtle phenotypes. Lastly, I plan to validate my pharmacologic experiments using constitutive Wnt and Notch receptor mutant mouse lines. Here are a few acknowledgements. Thank you to Dr. Blackshaw for giving me an opportunity to work in your lab. I'd also like to thank members of the Blackshaw lab, especially Leighton Duncan, for providing mentorship and guidance with my experiments. With that, can I answer any questions?